taking off the rest of our tops. And now we have a bunch of test tubes that have pipette tips and old culture in them. So I've grabbed the bleach. It's usually right over there by the sink. This is a mixture of 50% bleach, 50% DI water. And then we just give a little quick squirt to every single one of these tubes, just enough to kill all those cells that are already really unhealthy just because they've been sitting in this rack for a couple weeks. I just let a rack accumulate tubes until it fills up, and then I just kill them all. So these are all filled with bleach now, and I just take my tape and kind of wad it all together and toss that. I'm going to let these sit aside. I'm just going to let them sterilize for a little bit, and in the meantime I'm going to grab a 96 well block, which also has a bunch of culture. All these wells are full. So the easy way to sterilize this is, again, to take your bleach, start, and then just kind of go in a line. And you don't need that much bleach because a little bleach goes a long way. It's like you just want to hit every single well. Again, set that aside. And then here's a 24 well block. Just pull off the top. Fold it in half. Toss. And same thing. You're just going to shoot a little bit of bleach in. If you do a powerful stream, then the bleach will kind of mix with the culture and it'll just sterilize everything. So we're almost good. Nice strong jet there. So set that aside, and then our final thing to sterilize is going to be this flask with a bunch of culture in it. So we just squeeze it in there, and you can kind of see that there's a color change. It clears a little bit, gets a little bit bubbly, and then we just let them chill for a couple minutes. We're gonna pretend that they've already sterilized. I just put that bleach back there, and in the meantime we'll just cap everything up. So I'm gonna put all these tubes back, or all these tube tops back, and then we're gonna actually rinse all this bleach out. So we'll start with the easy ones. We'll take our 96 well, 24 well block, and our flask, and you're just gonna kind of dump them out right in the drain, and you want to get all that concentrated bleach stuff out of the way. So these have been pretty well bleached, so everything is pretty well dead in there. And then you want to turn on the jet. Now you want to be careful here, because if I just put this block straight in, the water's going to go down into that well and shoot back up, and it can get in your eyes, and that's no good. So what you want to do is you want to face the block away from you, and you want to kind of come at an angle so that if it does spray, it sprays in that direction. And you don't want to have the water too high. I'm actually going to put it up just a little bit, but I just kind of start there and then go make little lines up and down that block. So I'm going up and down those rows, and I empty it out, and that just kind of dilutes out any of that bleach that's in there, so that when it gets autoclaved and we reuse these blocks, we don't kill all of our culture with too much bleach. So I like to do it like 3 or 4 times. That's time number 3, number 4, and then to dry it, you just stick it on top of one of these skyscrapers. And we're going to do the same thing with that 96 well block. Now if you've only bleached a portion of it, then you can just kind of focus on the portion that you bleached. And it's always important to remember to wear your a lab coat and gloves for this portion, just because the bleach can mess up your clothes if you care. Either that or just wear rags. Well, you should always be wearing proper protective equipment in the lab. So the 96 well blocks are a little bit harder to rinse out, just because the surface tension of the water makes it difficult to get the jet of water in there. So you need to rinse that one a little bit more. And when you're trying to get rid of any of the liquids that are inside of the wells of that 96 well block, you need to give it a little bit more of a jolt so that the water shoots out. And again, put that right on top. Finally, we have our flask. This one's pretty easy. Just fill it up, empty it, fill it up, empty it. I like to do this about 5 times. That's 3, 4, 5. And this doesn't have to be a perfect washing, because these are all going to get washed in a machine washer. And then I just kind of put it upside down. We have quite a large accumulation of dishes here, it looks like. And then the final step is, or the final kind of lesson in this little cleaning demonstration is the test tubes. And these are kind of the trickiest, because oftentimes they have pipette tips or toothpicks inside of them. So you want to make sure that they all come out without breaking everything. So my method for this is I actually just start by trying to get rid of all the bleach. So in order to do that, I put my pipette or my tubes in the sink, put them upright, and then kind of tip it forward against the wall so that the tubes can't slink out of the tube. So if I didn't do that by the wall, you would see that the tubes start to move out. So I kind of use that wall to block it. Put it right side up, and then again I use the same sort of technique with the 24-volt block. And I turn on my jet, and I face it away from me, and I kind of try to go up and down those rows to fill them up with water. And I'm actually going to turn up the water just a little bit here. So you can see that they do start to shoot jets of water out. So you got to be careful. You don't want this to get into your eyes or on your clothes. Everything's dead in here, but bleach is still a little bit hard on you. So we've filled up these test tubes mostly with water. And we're just going to kind of do that same thing again. Pour them out, blocking the tubes from leaving the rack with that wall. And kind of the goal here is to not only get all of that bleach and the culture out of the tube, but you want to get all of the pipette tips out as well. Otherwise they might clog up the machine washer when we take them upstairs. And that's just no good. So I try to do this about 3 or 4 times. So just fill up all those tubes, and every time you do this, you're just diluting out all of that bleach and all that culture more and more and more. So that's time number 2. Just a couple more times and turn up that water. And you can see that I'm really, as I go up and down, just that stream of water is blasting out a lot of the bleach and a lot of the culture. And sometimes you even get the pipette tip or 2 shooting out of it. And that's good for right now. And then the final step is to just grab an empty test tube rack, which is going to be right here. Why I keep this over the sink. Stick it on the opposite end.
You missed the...
Oh, I missed the, yeah. And then we're going to flip it upside down so that they can drain. And then just shake it off a little bit, and then bring it and just stick it on top of one of these stacks over here.
Holy shit,
and we really need to do dishes. And then all of this is ready to go to the machine wash. It's been sterilized. It's going to get blasted with more detergent and water, and then we're going to autoclave it.